promising new research study may lead to improved and earlier detection of endometrial cancer.
The doctor who published this noteworthy study in PLOS Medicine is our guest on this week's Health Talk.
So stay tuned, we're up next.
Hello, I'm Dr. Eric Mazur.
And I'm Dr. Andrea Peterson.
Today on Health Talk, we're going to talk about endometrial cancer research with Dr. John Martignetti. John, thank you for joining us.
Thank you so much for
the
being here.
first time on the show, too.
Pleasure. Thank you.
So, John, your interest has been, you're primarily a researcher, even though you're both an MD and a PhD, and you've been studying endometrial cancer. Tell us, how did you get interested in that?
Sure. So, exactly as you say, my background is, in clinical medicine, but over the years we've really tried to develop a laboratory to really try improving diagnostics, understanding of diseases, really with the focus in genetics, using the power of genetics to get into that. And about 10 years ago, I was fortunate enough to meet Peter Dottino, who's the director of Gyn-Onc Services at Mount Sinai, and together we came, up with the idea of using our genetic framework that we've developed in the laboratory to study gynecologic cancers. And in particular, endometrial cancer, which we're talking about today, is an important cancer to study for a number of reasons. But if I could, I actually brought, I guess being a researcher, I brought slides.
Let me ask you,
Yeah.
how have you been studying? I mean,
Sure.
for folks at home who think about genetics, they think about fruit flies and Grendel Megger. No, I got that wrong. Get it right. Anyway, so now we have a whole bunch of people up on the screen, and you're using actually sophisticated techniques to look exactly at the structure of the gene.
Exactly, Eric, your point's spot on. In the sense that what we're able to do today is not just think about 70% of patients will respond this way, 30% won't, 10% will do this, 5%. Really, the goal of what people may have heard, this concept of personalized or precision medicine, is not just to treat everyone as the same individual, but really to find that one unique individual. So for an example, this slide where you have the woman in the red dress, it's the lady in the red dress, it's really to break down specifically, look at the tumor or look at their genetic information, sequence blood or tumor or other tissues to say, this is particularly unique about this patient's cancer. Not to lump, but really to get very granular and break down. And so,
and part of this is really because we now understand that endometrial cancer is not one disease, but is got a bunch of different genetic drivers, and they may behave very differently depending upon what the drivers are.
Exactly, and this is what we're seeing for all cancers,
And that's one of the problems that I see is, we talk about cancer as this monolithic disease. It's sort of been the way we've been talking about the cure for cancer, and it's really like saying, the cure for all infectious disease. There's just so many, they're all different diseases.
Different.
Right, and indeed, it's not just the cancer that's different, but how people respond to those cancers. People come in with different health levels and different degrees of resistance or resilience. So, it's very important now for us to be able to break down the cancer. And exactly as you say, we realize now that many cancers are not just ovarian, endometrial, leukemia. There's specific subsets. It's important not just...
And subsets.
Exactly.
It really, it's almost like a babushka doll from the Russians. You just keep... You think you've got it understood, you open it up and there's another one there and you've got to keep... it's really sort of striking.
It's important, exactly, and it's super striking because it has clinical implications. Yeah. Patients may respond to drugs differently, better or worse. Patients may have different outcomes, based on the genetics.
I don't want to dominate the conversation, but I just, maybe for the folks at home, they may not realize, since the Human Genome Project isn't that old, that we now have the capacity to sequence virtually, or many, if not most, of the genes in an active cancer in an individual patient in less than 10 years. You just say the technology has really exploded. So say a few words about that.
Yeah, so I can tell you. So, I was fortunate enough to be in one of these laboratories where people were first starting to do these large-scale sequencing projects. And they involve really quite frighteningly, using a radioactive material, taking a mouth pipette with a long rubber tube, and, and just kind of pipetting these into these, glass plates and sequencing. Now this is done all automated, and the scale is incredible. What would take us, for example, we could sequence 350 nucleotides in a day. Now you can sequence 350,000 in minutes. It's an incredible scale. It's now really the problem is not the technology to sequence, but the technology to decode all that information and make sense of it. And it's so important, again, for people listening, patients and families and people who are just interested. It really is, we're at that convergence of technologies, for sequencing, the chemistries that go into this, the bioinformatics, the math skills, but also really the clinicians that have to really help explain and decode what goes on in this. And I think we're really at that convergence point now.
And I think that's such an important point. Maybe we can take a step back too to the person level and talk a little bit about endometrial cancer, and maybe you can say a few words about who gets endometrial cancer and why it's such an important cancer to be dedicating time and resources to.
Sure, and I brought a couple of slides again. Again, being a researcher, we have to have slides. So endometrial cancer, currently this year, it's expected we'll have about 55,000 women in the U.S.
Say, what's the endometrium for folks at home?
Sure. So, the endometrium is the uterus. It's the other name for the uterus. So this is a gynecologic cancer. It's found in women, and it's a cancer that develops over lifetime. As you get older, your risks go up. And the symptoms that you might have, I've listed a couple of the symptoms. So primarily would be things like postmenopausal bleeding. Once your bleeding has stopped, if you've been 53, 54, 50, 45, and you've stopped your periods, all of a sudden you may have bleeding again. You may have pain. Doc may identify some type of a polyp or a fibroid in your uterus. These are all symptoms of endometrial cancer. And if you look at that picture that I've shown you there where it lists out...
Before we scare too many people home, fibroids are pretty normal and
exactly.
are not cancer.
Exactly. It's a symptom. And again, that's exactly why we did the research, Eric. So you're spot on with the question, because the physician, the patient, is confounded with these things. So one of the primary drivers for a woman to go to their gynecologist is having postmenopausal bleeding. It's only a small fraction of those women that actually have cancer. The other things are endocrine related. There's a lot of other reasons, and those are usually the main reasons. The problem becomes, and why we got interested in the research is, that 55,000 women that'll have endometrial cancer this year, the numbers are rising. It's already been, essentially hypothesized that by 2030, so really in about 10, 15 years, the number of cases will actually increase, and it'll actually be more common than colorectal cancer.
Why is that?
So probably a lot of reasons. Probably the two main drivers are use of estrogens and obesity. So more weight that there is, essentially the hormones that are released with being obese contribute to the growth of endometrial cancer. So those are probably the two main drivers. So now you've got this, where you have the suspicion and the diagnosis, right? Because now you go to the physician says, I suspect this or is thinking this, and there are a couple of things you can do to have the diagnosis made. You can have ultrasounds, you can have a physical exam, you can have CT or PET imaging, nuclear imaging. But ultimately, the gold standard is bringing the patient to the operating room, putting them under anesthesia, and doing what's called a hysteroscopy. Now, the problems with that is you've got to go to the operating room, you've got to have anesthesia. It's a big deal.
And a hysteroscopy is they're actually taking a probe and looking up into through the cervix into the inside of the uterus.
Exactly, and we have a slide on that, actually.
Yeah,
so the next slide that we have shows the hysteroscope. So essentially, it's the surgeon's tool to look into the uterus, the sac, the uterus, the endometrial lining that's there. So the surgeon looks up in there and is able to, and what I've shown on the slide on the bottom right-hand corner is essentially what the surgeon sees. They're looking in there. Now, again, the worrisome thing from a research side and where we'd like to do better than what's currently available is, again, you have issues of going to the operating room, anesthesia, the pain, discomfort, loss of time, and ultimately what happens, it's a subjective test. When the hysteroscopy is done, two things are done, and I'll tell you the second thing first. But the second thing is the surgeon actually takes a sampling of tissue. tissue. It's a DNC, a dilation and curettage. So the uterus is expanded, and then some of the tissue is scraped. That tissue gets sent to the pathologist, and the pathologist has to look and say, I see or I do not see cancer. There's no special stains. There's no special tests. It really is the experience and the guidance of that pathologist to look and see.
They're looking at the tissue under the microscope.
Under the microscope.
And traditionally, that was the way that all cancer diagnoses used to be made.
Exactly, exactly. So that's, one of the things that we're trying to improve upon, because about 15% of women that have the diagnosis, it's either missed diagnosis or there's a variation in what happens, what's expected on that. And so having a molecular test would then make everyone equal across the world. In other words, the experience, would always be gained from the pathologist looking, but they would have a backup, a molecular test. And in some places where you're not as lucky, let's say here, Danbury, we've got specialized pathologists, but maybe if you're in a smaller place or a place that doesn't have that experience, again, having a molecular test kind of levels the playing field, and it's best for the patient.
So two pieces of tissue that may look similar just by eye or on the microscope may actually be very different on a genetic or molecular level, and that can have real implications for how that cancer is going to act in that particular woman and what kinds of treatments might work best for her.
Exactly, and exactly Tarek's point when we first started speaking, exactly as you say, Andrea, that the pathologist's job is to say, yes, no cancer, and then they can grade it into two types, an aggressive form and a less aggressive form. The molecular really should be able to add to that and say, yes, I believe that. Maybe it's not one, not two, but maybe it's three, or it's a mix. And so that's always the
And do you have a
hope.
final graphic that summarizes this?
So as I look up to see whether or not we have the graphic. So indeed, this is the test that we did. So as I said to you, the surgeon does one other thing. In addition to taking that scraping of tissue, they actually just essentially blow in some salt water, because that opens up the cavity and allows them to see in. What we did in this study was we combined the gold standard testing of the surgeon taking patients to the operating room. We did not know a diagnosis, they did not know the diagnosis, but these were 100 women that went to the operating room to have the procedure done, to have a definitive diagnosis made. We asked the surgeons and the patients, can we take a sampling of that salt water that you guys are just essentially opening up the cavity and would be normally considered waste, can we bring that back to the laboratory and sequence it? Look for the DNA that's there, look for cells that may be there, and then apply all this new technology to see what we find. And then at the end of the study, after we've looked at these 100 women, compare results.
Now, you didn't look at all genes. You looked at specific genes that you knew were associated with endometrial cancer, right?
Exactly, and it's a very good point, because indeed one could do that if you had all the money in the world and if you had all the time in the world, but if you can try focusing on some genes that are more informative, that really should make these things go in real time, and indeed that's what we were able to do, Eric. So exactly, we had two panels that we had devised. One panel had 56 genes on it, and one panel had 12 genes. And our bioinformaticians and statisticians, in particular Boris Riva, had looked at all of the sequencing that had been done, looking at all the genes, and he said, I believe these to be 12 most informative genes. And so that's actually what we did. So really, if people remember now that slide that we had, we took the saline, we came back to the laboratory, separated it, and then sequenced just those 12 genes, and then had essentially a printout at the end. And that really is the research.
And so when we come back, we'll hear the results of that research.
Perfect time
really
for our
fascinating.
break.
Sort of, this is the edge of medical science where we are.
Absolutely.
Really looking at the genes and tying them into the patient and whether or not they have cancer.
We do have to take a short break though, so stay tuned. We'll be back.
We'll be back. Bye-bye. Hi, Andrea and I are back with Dr. John Martignetti. We're hearing about this fascinating study you did looking at the exfoliated cells in the uterus when women were being otherwise clinically evaluated appropriately for endometrial carcinoma. So there was really no extra thing done to these women. I presume they all provided permission to use the saline washes, but there was no extra discomfort, even extra tissue samples. So, so tell us then a little bit what you found.
What did you find?
Sure. I have yet another slide,
Excellent.
surprisingly. I have one more slide. So what I have on this
Here
set
we
of
are.
slides here is this actually, so the patient, this is really a chance for them to see what their material looks like. As I told you, remember, the surgeon will take a little piece of this tissue when they look in. This gets sent up to pathology, and this is actually what the pathologist now reads.
And just to orient our
Yep.
Viewers.
our viewers, I was going to say patients, each one of those orange things with an extension is a slide.
Exactly.
The orange thing is a label, and those purple areas are the tissue that's been stained. And I guess each one of those is maybe the size of a dime or something like that, just to put a size on them.
Exactly.
It's a small amount of tissue.
Exactly. And so that would all have been generated from one patient. And now it's the pathologist's job to look under the microscope and really deduce, from their experience, what is going on. Now, as we're looking at it again, so it's a series of rectangles, that's all from one patient. On that second row, that there's one gap, people probably easiest to see, that there's one gap there of slides, and the one just to the right of it. For people who are just really sitting right up close to the television set, they might be able to see that there's a tiny black dot next to the slide, that the pathologist with a little felt tip pen put a black dot. That's one of the tumors that we were able to identify using the genetic technique.
Just to make it looks like that's pretty easy to miss, because if you slice on either side of it, you may miss that cancer altogether.
Exactly. And so and that's not necessarily the pathologist's fault, because
No.
exactly as
Right.
you say, depending on how that section is cut. And so there is a little bit of luck involved in it. And that's what we want to take out of that. We want to get rid of that luck piece. But, oh, I'm sorry, Andrew.
No.
But to say that little black dot, for just people who are viewing at home, that if you have a fine tip point at home on a pen, that's really what that looks like. That is the size. So that's the power of what we could find.
And so what you're saying is that was a woman whose cancer was found on that pathologic slide, but you also were able to find the DNA from that cancer in the saline that you analyzed.
And so, there are two major results that we think from this research that we did. The first is, again, these were women who were coming in for a diagnosis. We didn't know, the surgeon didn't know, the woman didn't know. 7 of the women were diagnosed as having endometrial cancer. Early stage, so stage 1A, grade 1. So this is the earliest type of endometrial cancer. And as I showed in those slides, these were microscopic, and I think it's fair to say they were microscopic. The 7 women that were diagnosed as having endometrial cancer, of those 100, all 7 were detected by the genetic test. We found mutations in that saline wash. So that's really the first time that it's been done in a prospective, in other words, looking forward way, that the technology said we can identify cancer just as well as the pathologist.
And you mentioned to us before the show, you know that it's the DNA from the cancer because you actually went back, looked at the cancer, and matched the two
Very
samples
important.
of DNA.
Exactly, because again, one, you'd hate to think it's an error, and errors always do occur. We have some type of technology error, but indeed, exactly as you guys had asked, they matched exactly. So that shows, it shows that whatever DNA we picked up was really being shed by the tumor. So on that part of the research, the hope is, and it really does, kind of move the ball forward, that we think we can move towards a screening test. Because again, there are no screening tests for endometrial cancer. A woman has to develop symptoms, which means that the tumor has gotten to be a particular size. If we could step that back, we hope then women can get treated easier in the sense of what surgery has to be done and their eventual outcome. Because again, generally, if you catch a cancer earlier, you do better. That was the first part.
But before you
of the
even leave that, I think
Yeah.
that also may help change the way we think of endometrial cancer. I know that when I was caring for patients with tamoxifen, which is a anti-cancer drug, but it's got some estrogenic activity, we used to do an ultrasound to look at the endometrial stripe to see whether it was thickening, to see as an early sign of cancer. This is a much different and perhaps more specific way to look at those early changes of cancer. We were just looking at the image on an ultrasound.
And exactly, I think what the technology gives us, and I think we realize this more and more as we do these studies. If you were a doc maybe 100 years ago or 200 years ago, right, the tools you had were essentially your ability to listen, look, feel. X-rays were developed, and now you could listen, look, feel, and you could actually see inside of a body. 50 years ago, 20 years ago, as new radiology techniques, now you could do CT scans, PET scans, MRIs. You had really an unparalleled way of being able to look at your patient and understand. And probably a lot of times, though it doesn't get reported in the literature, and we don't learn that in medical school, is probably there was a lot of head scratching going on, like what does this mean for the patient, right? And so only time and experience gave that. That's what the technology is doing now, genetics. It's again affording you an insight into something you didn't even know was there before.
But it's from what I understand, this is still not ready for prime time as a screening test because you had a large number of women who did not have cancer. As I recall, it was 51 women, I think, who did not have cancer, but did have some of these cancer genes that you detected.
Exactly.
And so, I think, what are the implications of that?
So-
Because I think there are lots of them.
Yeah, and it's very important, exactly. So for those who are listening, again, this is not a genetic test that someone should be rushing out and asking about. This is something in the future, as is much research. And, clearly we're now building upon this. So we're now trying to do a 1,000-woman study where we learn more because, exactly as you say, the 7 women that had cancer, we detected their mutations. But unexpectedly, half of the women in our study also had mutations. Now, that really is unexpected because, if you open any cancer genetics textbook, if you sit in on any lecture in cancer genetics, if I told someone, or you heard the professor or the lecturer saying, there's a mutation in this gene as we identified in our patients, your leap of faith would say, this patient has cancer or is developing the steps towards cancer. But that's not what we saw. We had half of the women had these very strong, very well characterized, very well described genetic mutations, but they didn't have clinical evidence of cancer. So what does that mean? That really is the next important thing.
Does this change the way we view cancer, or is potentially, I guess, changes the way we view?
I'd like to think so. And of course, as you know, I've got 2 slides
Well, here,
for
we've got
that.
one up.
Exactly. So, generally when you think about cancer, you say, okay, it's a series of steps, genetic mutations that lead a cell astray, that lead to the development of cancer. And so, if you think about it, it's maybe like a doorway where you're just on the precipice, you've gotten all these steps that have brought you to the arch of this doorway, and the next step in, your cancer. Now, that works if you think of it as a hallway, but the reality and what this research shows, so if we look at another slide that I prepared, which I've stolen from, I think, Verizon and Fios, it's essentially, if you're on the other side of that wall, and you have all these mutations, yes, they may all be rushing towards the possible development of cancer, but not all of them are getting through that door. And that's the really exciting thing from the research, and I think from the clinical point of this, because it's telling us that these mutations arise in patients, but somehow they do not develop into cancer. Why is that? And if we could learn that, can we do better on cancer prevention? That would really be the next goal.
What are some of the possibilities about why some of these may not turn into cancer?
Yeah, Andrea, so it's a great question, and it's really the million-dollar question. So I think a lot of things that we're thinking about would be, we could continue along, what we've always thought about. Maybe there's some other steps in the genetics that we haven't appreciated yet. So maybe there's some other mutations beyond the ones that we've looked for that could be involved. It could also be, again, as we started the segment, talking about things from outside that cancer cell. It could be someone's immune system, somehow detecting what's beginning and putting the brakes on it. Could be something like that. That's what we really need to learn, because again, if we can learn what it is, then maybe we can apply it and we might be able to prevent more cancers.
And unfortunately, that's all we have time for. There's so many fascinating implications of this in younger people. We could go talking forever. But we're going to keep you here to answer the question and come back after a few messages.
Don't go away. This week's health question is, what's the goal of the new Laboratory for Translational Research at Western Connecticut Health Network?
And you're the head of that, are you not?
Yes, I am.
So tell us, what's the goal?
So the goal really, it's really to encompass what we were speaking about today, really the bigger picture of that, to try bringing this concept of precision or personalized medicine to the patients that we care for at Western Connecticut and really, and Mount Sinai. And, it's not just the technology development, as we've been speaking, and you guys have pointed out exactly. It's really the clinical understanding that goes with that, and hopefully, bringing forth new questions for us to even start thinking about. So the goal, to bring precision care to our patients. I think the other parts of it will be understanding.
And the word translational there really indicates the bridge between what goes on in the research laboratory and the patient. Exactly. And how we can really use the science to help real people who are battling diseases.
Exactly. That is the ultimate goal.
It's amazing that cancer is turning out to be much more complicated than we ever expected. Again, I remember back when I was in training, they talked to an initiator gene and a promoter. They were talking about a two genetic model or a two-hit model for cancer. And now we know that it may involve dozens or hundreds, and maybe there were suppressor genes that interact. It's, maybe you could just say a couple words about that, but the complexity really is extraordinary.
I have a friend, a beloved friend, who's an X-ray crystallographer, which always to me was like, wow, and he always, he's always said to me, the devil is in the details, right? And that's exactly what it is. As we start studying these diseases more and more, we have new technologies to really break down and really understand better. We keep finding new avenues of
understanding. We keep unwrapping the onion, but there's still onion left.
We're so glad you're at Western Connecticut to help with this research and help all our patients.
This was a fascinating discussion. I hope the viewers at home were able to appreciate what we were talking about. So if you at home, though, have a question you'd like to ask on Health Talk, please contact Andrea and me at HealthTalk@norwalkhealth.org.
We'd like to thank our guest today, Dr. John Martignetti, for coming on our show with great information.
And we'll see you next week on Health Talk.
Be well.